What are the three layers formation we have seen in the histogenesis? We have seen the formation of matrix layer, mantle cell layer and marginal cell layer. These are the three cell layers we have discussed in the last session. Okay, we'll start with the development of spinal cord and also hindbrain in this session. Okay. Okay, so in the histogenesis we have seen, we have the, what is this structure called? The cavity like structure, it is called as neurocoel. It is called as neurocoel. Okay, and this cavity is lined by your pseudostratified columnar epithelial. These two structures will together form the matrix layer, matrix cell layer. And the cells present in this layer will undergo proliferation and forms the one more layer, that is called the mantle layer. Okay, it is called as mantle cell layer. And the mantle cell layer consists of your spongioblast cells and neuroblast cells. Okay, again after this we can see one more layer, that is called as marginal cell layer. It is called as marginal cell layer, and the marginal cell layer consists of axons of the neuroblast and also the matured spongioblast cells. Okay, so keeping this cell layer structure as a basic or a reference, we have to study the development of your spinal cord. Okay, your neurocoel is going to develop as the central canal of the spinal cord. Okay, so the neurocoel is going to develop as the central canal. Okay, and your mantle cell layer is here, is the future which matter, grey matter or white matter? Grey matter. Okay, and the marginal cell layer is the future white matter of the spinal cord. Okay, so this is the central canal of your spinal cord, and this part we call it as the roof plate, and this part we call it as the floor plate. On either side of this structure we have the lateral walls. Okay, this is the lateral wall. Stop talking. Your roof plate and your floor plate are thin in structure, whereas your lateral plate is thick in structure. These points we have already discussed. Okay, between the dorsal part of this is the dorsal part of your spinal cord, and this is the ventral part of your spinal cord. Okay, between the dorsal and ventral part of your spinal cord we have a sulcus, this structure. Okay, this structure what you are seeing here, no, this is the sulcus. This sulcus is called as sulcus limitans. Okay, this structure is called as the sulcus limitans, and this sulcus limitans will divide your lateral wall into dorsal part and ventral part. Okay, the dorsal part is named as alar lamina, and the ventral part is named as basal lamina. This is named as basal lamina. Okay, any doubts in this? Any doubts? Keeping the sulcus limitans as the reference, we divide the lateral wall of the spinal cord into two parts, dorsal part and ventral part. The dorsal part is also known as alar lamina, and the ventral part is also referred as basal lamina. Okay, your alar lamina is sensory in function or motor in function? Sensory. Okay, this is sensory in function, and the basal lamina is motor in function. Okay, so if this is sensory in function, here in this area, that is in the dorsal part, we can see the afferent columns or efferent columns. Afferent columns or efferent columns. Which columns we can see? A or E? A. Okay. We can see the afferent columns, and in motor area we can see the efferent columns. Okay, is it right? Sensor, motory, afferent, efferent, everything is right? Sure, everyone? Okay, fine. So, your spinal cord only have the general columns, okay? Remember this point. Remember this point, your spinal cord only have general columns. Whereas in your brain structure, along with the general columns, we can also see some special columns, okay? So, in the spinal cord part, we can see 4 columns, 2 on the alar lamina and 2 on the motor part or on the basal lamina, okay? Can you name the columns? So, this is general, sensory or visceral? This is general, sensory, afferent column, and this is general, visceral, afferent column. Okay. In the basal part, this is the general, visceral, efferent column, and this is the general, somatic, afferent column. Is it clear? Any doubts? Any doubts in this? So, in your spinal cord, we can see total 4 columns, okay, and all the 4 columns are general columns. We cannot see any special columns in the spinal cord, okay. The first one is general somatic afferent column and general visceral afferent column, which is present in your alar lamina part, and the other 2 are general visceral afferent column and general sensory afferent column, which we can be seen in the basal lamina part, okay. Your somatic columns, I'll write it here, somatic columns are present throughout the spinal cord, okay, throughout the spinal cord. And your visceral columns are present only in the thoracic region, lumbar region and sacral regions. MCQ questions, okay. The somatic columns, this one and this one can be seen throughout the spinal cord, whereas the visceral columns can be seen only in the thoracic region, lumbar region and sacral region. The concept of knowing about the columns of your spinal cord is called functional components of the spinal cord. Okay, whatever the diagram I have written here, no, this whole concept is called as the functional component of spinal cord. Short note question. Explain briefly the functional components of spinal cord. You have to write this diagram and you have to explain all the structures. Mainly you have to mention about sulcus limitans and alar lamina, basal lamina. Along with that, mention sensory, alar lamina is sensory in function and basal lamina is motor in function. Later go for all the 4 nuclei or columns, okay. Any doubts in this? Any doubts in the functional component? These points explain the development of your spinal cord, and this whole diagram is for the functional components of the spinal cord. Any doubts? So, shall we go to the next topic? Okay. Next topic is called positional changes of the spinal cord. So, what do you mean by the positional changes of the spinal cord? Gross anatomy of spinal cord is finished. No, not yet. What they are discussing in gross part? Meninges and CSF. Okay, fine. Okay. The next concept is called positional changes of spinal cord. Okay. By the end of 8th week, your spinal cord and your vertebral column, this is the spinal cord, okay, and this is the vertebral column, okay. These two are present at the same level, okay, they are present at the same level. As the development continues, your spinal cord will remain the same, but only your vertebral column will get extended like this. So this change can be seen at the 24th week. Okay. So here the upper part will be at the same level, only we can see changes in the lower part or at the caudal end of your spinal cord. Remember this point. So here it can be seen at the level of which vertebrae? It can be seen at the level of which vertebrae? It can be seen at the level of S1 vertebrae. Okay. So at the time of birth, your spinal cord will present at the level of L3. Okay, it is present at the level of L3. After the birth, we'll check for the adult position. This is the final position we have to consider. Okay, so in the adult position, your spinal cord is present at the level of L1. Okay, it is present at the level of L1. This is spinal cord. So once the development of your spinal cord takes place, by the end of 8th week, if we check the position of your spinal cord and vertebral column, they are present equally or at the same level. Okay. So after few development or changes, if we check the position of these two structures, by the 25th week, your spinal cord is present at the level of S1 vertebrae. Okay. At the stage of birth or at the time of birth, your spinal cord is present at the level of L3 vertebrae, and the adult position, that is the final position, we can see the spinal cord at the level of L1 vertebrae. Okay. What about the spinal nerves which is present along with your vertebral column? So when the development takes place, each and every spinal nerve is present like this. So what happens when the positional changes takes place? So whatever the spinal nerves that present below the L1, that is your lumbar and which one? Sacral and present here will move in the downward direction and also towards the lateral side like this. Okay. Here your vertebral column is there, it will move like this. Okay. The position or a point where all the spinal nerves are held, from there, this is the point where all the spinal nerves are held. Below this, all these three nerves will move in the downward direction and also towards the lateral side. So this point is called as Cauda, C-A-U-D-A, Cauda equina. Okay. This point is called as Cauda equina. You can see that gross structure in the dissection. Okay. So this is all about the positional changes of your spinal cord. You should know from 8th week, 24th week and at the time of birth, then the adult position or final position. Along with that, you have to know about one important point, that is Cauda equina. Okay. This finishes your development of spinal cord. So any doubts in development of spinal cord? Any doubts? Okay, go through your notes once. Take 2 minutes and see the notes or the points, whatever you have written. If you have any doubts, we can discuss. Okay. The development of spinal cord. Matrix zone is thick and lines the enclosed cavity, that is called the neurosil. Its numerous cells undergoing mitosis produces neuroblast cells and also spongioblast cells. The former develops into your neurons and later into neuroglial cells. Okay. The neuroblast migrate to the adjacent mantle cell zone or cell layer zone. It is going to develop as the future grey matter. And their axons enter the external marginal zone and it will develop into your white matter of the spinal cord. Okay. Once histogenesis is complete, the remaining matrix cells differentiate into ependymal cells that lines your central canal part. Development of the functional columns. On cross section, the cavity of neural tube appears like a vertical slit. So that is the reason we will mention here as the roof, floor and also study the lateral walls part. Due to the formation of vertical slit, the dorsal and ventral walls of the neural tube remains thin and they are called as roof and the floor plates respectively. The lateral walls of the neural tube get thickened. On each side, the lateral wall of neural tube is demarcated into dorsal and ventral regions by an inner longitudinal sulcus, that is called as the sulcus limitans. Any doubts? The cells of dorsal region is named as alar lamina, which are functionally afferent or sensory, while those of basal lamina are efferent or motor. The two efferent columns of alar lamina receives axons from the dorsal root ganglia. They are general somatic afferent column, general visceral afferent columns. The two efferent columns of basal lamina give rise to your motor fibers. Those are general visceral afferent column and general somatic efferent columns. Remember, the somatic columns is present on the outer part, viscerals are present in the middle part. So the somatic will supply to or present throughout your spinal cord, whereas the viscerals are restricted only to the thoracic, lumbar and sacral regions. Okay. So this is the diagram for the roof plate and lateral walls. The B diagram is to show the three cell layers. Okay. You can see the formation of sulcus limitans. Once the formation of sulcus limitans takes place, it is divided into alar lamina and basal lamina part. Okay, and this is the adult structure of your spinal cord. I hope you have seen the similar structure in your histology. So I am not going to explain it here. It is already finished. But you should know which is the basic structures from which the adult structures are derived. Okay. So your central canal is derived from neurosil, and what is this? This is the grey matter part and it is derived from your mantle cell layer, and this is the outer white matter. It is derived from your marginals. Okay. This is the diagram for the functional components. Any doubts in the diagram? Where these nerves are running into? Can you see the nerves here? Where they are running? Where exactly those nerves are running? Okay, this is the column area, right? From here, can you see this structure? The black lined structure, what you call them? Anterior and Roots. Okay, fine. So where these roots are going? Okay. Hmm. It will divide into two ramus. Okay, fine. What are those ramus or called? Anti Anterior and posterior. So what is the function of ramus? Okay. Back muscles. So you are telling me no roots are going into your head region, only to the lower body region, like your limbs part and thorax. Uh, okay. Others? Okay, fine. Next, we have the positional changes of the developing spinal cord. At week 8, the length of the spinal cord and the vertebral column are equal. The spinal cord extends along entire length of the vertebral canal and the spinal nerve exit intervertebral foramen at the level of their origin points. Due to the differential growth of the cord and vertebral column, the intervertebral foramen do not remain at the level of the spinal nerves. In order to exit this corresponding intervertebral foramen, due to the recession of the spinal cord, the spinal nerves are forced to go down in the oblique direction. What do you mean by recession of the spinal cord? Okay, tell me what do you mean by recession at least. Short. Okay. Any other answers? Falling behind. Okay. Others? Second row girls. What do you mean by recession of spinal cord? So by the week 24, the lower end of the spinal cord ends at the level of S1 vertebrae. At the birth time, the lower end of the spinal cord ends at the level of L3 vertebrae. In adults, that is the final position due to further recession of the cord. Again, they are mentioning about the further recession of the cord. The lower end of the spinal cord ends at the lower border of your L1 vertebrae. The nerve roots that descends below the lower end of the spinal cord, and that point is called as the conus medullaris, surround thin thread-like prolongation of pia mater from tip of the conus medullaris to form the cauda equina. All these structures what they have mentioned, no, you can see them in the dissection part. Okay, right now just try to remember the names. Okay, this is the picture for the positional changes of your spinal cord. Compare all the four structures with the diagram what I have written. They have shown only the caudal part of the spinal cord, okay? They are not showing the cranial part, only the caudal part along with your conus medullaris and your cauda equina part. Don't get confused, okay? Once check in other textbook also, if you have the picture from cranial to caudal, keep that picture as your reference. It will be more convenient to study. Okay. Any doubts in this? So that finishes your development of spinal cord. You have to write about the histogenesis part, later the adult derivative of that histogenesis part, followed by functional component, positional changes and clinical anatomy. For the clinical anatomy, you have to mention about the spina bifida part. Okay. Any doubts? Okay. Next, we will go to the development of your hindbrain. So, tell me the brain vesicles part. How the development of brain vesicles takes place? In which week we can see the development of brain vesicles? In which week we can see the development of brain vesicles? What are the two types of brain vesicles we studied? Open your notes or textbook and then answer at least. When we can see the development of brain vesicle and what are the different types of brain vesicles we have studied. Stop talking. So in which week we can see the development of brain vesicle? We can see the development of brain vesicles in the 3rd and 4th week. Okay, so first we can see the neural tube getting or forming your primary brain vesicle. Those are Prosencephalon, Mesencephalon and Rhombencephalon. From the primary brain vesicles, we can see the formation of secondary brain vesicles. What are the secondary brain vesicles? What are the secondary brain vesicles? This is the telencephalon followed by diencephalon, then we have mesencephalon and next metencephalon and myelencephalon. This is going to develop as your spinal cord. Now we have already seen the development of spinal cord, so we will go from the below to the above structures. Okay, now we have to study this part. So that is your metencephalon and myelencephalon. Your metencephalon will give rise to which structures? Pons, okay, pons, and it will give rise to pons and cerebellum. Okay, and your myelencephalon will give rise to your medulla oblongata part. So the development of medulla oblongata is very similar to the development of your spinal cord. Okay, any doubts? Okay, we'll start with the development of your medulla oblongata. Okay. So now if I take the cross section of your myelencephalon, we can see the 3 cell layers similar to your which part? Similar to the spinal cord part. Okay. So now the outermost one is marginal one, and next we have mantle layer followed by the matrix layer. Whatever the cavity present here, no, the neurocele part. In your myelencephalon, the neurocele part is going to develop as your which ventricle? If you remember about the development of ventricular system, I mentioned whatever the cavity present from here to here is going to develop as the different structures, right? So which ventricle we can see in your hindbrain region? It is going to develop as your 4th ventricle. In case of spinal cord, it will be developed as your central canal. Okay, remember this point here, the neurocele is going to develop as the 4th ventricle. To study the development of your medulla oblongata, we have to concentrate on the mantle cell layer. Okay. So during the process of development, the ventral part and dorsal part division is already present. We all know there is sulcus limitans. It is dividing your lateral wall into dorsal part and ventral part. So the ventral part, that means this part, ventral part is going to develop rapidly or fast. As a result, only the ventral part will get expanded in size like this, okay? Whereas your dorsal part shows slow growth compared to the ventral part, so it will remain the same in the size like this, okay? Okay, so this is the dorsal part now, which is similar to the structure of your spinal cord. So we call it as alar lamina. For alar lamina and basal lamina, in few textbooks they even mentioned as alar plate and basal plate. Okay, you can remember like that also. And this is your basal lamina part. So due to the expansion of the ventral part, this cavity present here will be compressed in the ventral region, okay? And this, whatever the sulcus it is forming here, it is called as sulcus limitans. Sulcus limitans. Similar to your spinal cord structures only. And this cavity, whatever is present, is going to develop as the fourth ventricle. Okay. Any doubts? Any doubts in this? Okay. Then again we'll come back to this structure. Remember, this will be your roof plate and this will be your floor plate. Okay. So now we can see some changes in the roof plate due to the flexure of your brain part. Okay. I'll write it here. There will be some changes in the roof plate due to the flexure or formation of flexure, due to the formation of flexure. So tell me, what is flexure and how many types of flexures we have studied? Flexure is basically the bending of your developing brain part, because during the development in the initial stage, that is at the week of 3rd and 4th, your skull area is not spatial enough to occupy the developing brain part. So the brain part will undergo few changes and it will show us 4 types of flexures. What are those 4 types of flexure? We can, anyone please? Cervical flexure, Pontine flexure, cervical flexure, Pontine flexure, and cephalic. What cephalic? Any particular name? Mesencephalic and Rhomben. Okay, fine. Now tell me, in which area we can see all these flexures? Okay, between the spinal cord and your Rhombencephalon, this part we can see one flexure, that is called as the cervical flexure. Okay, next Pontine flexure. Where we can see Pontine flexure? Pontine flexure takes place between your it happens in the Rhombencephalon. So this is the Rhombencephalon part. When the flexure takes place, it will give us 2 cephalons. This is the Metencephalon and this is the Myelencephalon. This is the Pontine flexure. Okay. So what about the other 2 flexures? Mesencephalic flexure, where we can see that It takes place in the mesencephalon area. What about this area? Is there any flexure? Cervical flexure, pontine flexure, mesencephalic flexure. The last one is rhombencephalic flexure. Okay, fine. Now we want to see the pontine flexure. Okay. So during this development, along with the formation of all these layers and sulcus, your flexure will also takes place. Once the flexure takes place, this is how the structure will look. Okay, so this is your roof plate. So once the pontine flexure occurs, your roof plate will become thin and it will get elongated. Okay, here if you see, this is the roof plate structure. Okay, once the pontine flexure is taking place, it is going to become a very thin structure and it will also get elongated on the either side. Okay, so due to the elongation of this roof part, this part, your alar lamina is going to push towards the lateral side. Okay, so now this is the alar lamina part. Okay, your alar lamina is pushed towards the dorsolateral side. Okay, alar lamina. And your basal lamina will remain the same. So for the development of your hindbrain, right now we are discussing about the development of your medulla oblongata. To study the development of medulla oblongata, we have to concentrate more on the mantle zone. Okay. So once we start studying about the mantle zone, we have the alar lamina, basal lamina and sulcus limitans. Okay. So in the roof plate, due to the pontine flexure, there will be certain changes. Your roof plate is going to be thin and it will become extended on the both the side. As a result, your alar lamina will be pushed towards the dorsolateral side. Okay. Now remember, this is only about the mantle cell layer. Next to that, we have marginal cell layer. Okay. So whatever the cells present in your alar lamina will now start moving towards both the direction. One in the downward direction and one in the upward direction. Okay. I'll write one more diagram. Shall I clean this? Finished? Okay. So this is your basal lamina and this is your alar lamina. And this is the roof plate. Okay. So now, not all the cells present in your alar lamina, but the few cells present in your alar lamina will start moving to your next layer region. That is the marginal layer. Cell layer. Okay. Few will move in the downward direction and few cells will move in the upward direction. Okay. The cells which move in the downward direction will give rise to inferior olivary nucleus. Okay. And the cells which move in the upward direction is moving towards your which part? Metencephalon part. So your metencephalon part is going to give rise for your pons and cerebellum, right? So this will give rise to your pontine nucleus. Okay, the cells, that is the lateral side of the cells present in our alar lamina, will move in both the direction, upward and downward direction. The cells which move in the downward direction will give rise to inferior olivary nucleus, and the cells which move in the upward direction is giving rise to your pontine nucleus. Okay, so this process of cells moving from your pontine region to your medulla oblongata region is called as what expansion? It is called as what expansion? The name itself is telling it is the cells are moving from your pontine region to your medulla oblongata region. So what that expansion is called as? Okay, everyone just go and check once what this expansion is called as. In the next session, I'll discuss about it once again, okay? So, any doubts in this? Any doubts? No doubts? Okay. Next, again, similar to the concept of functional components of your spinal cord, even in the medulla oblongata, we have functional components of the medulla oblongata, okay? So, in the basal region, we can see 3 sets of functional components, whereas in the alar region or lamina, we can see the 4 sets of functional components in the medulla oblongata. Okay, I'll write it again. Here we can see 3 sets of functional component, and here we can see 4 sets of functional components. Can you name them? Alar lamina means it is sensory in function. So we call it as afferent columns. So there are 4 columns. Can you name them? Remember, in your brain region we have general general columns plus special columns. Okay, can you name them now? We have general, somatic, afferent column and special somatic afferent column. And next we have general, visceral afferent column, stop talking, and special visceral afferent column. Okay, next we have basal lamina. Basal lamina means motor in function, and we name the columns here as afferent columns. But here we can see only 3 columns, okay. The first one is somatic columns, somatic afferent columns. The second one is general, visceral afferent columns, and the third one is special visceral afferent column. Third one, fourth. These are the total 7 columns of your developing hindbrain. Any doubts in this? Okay, once go through the notes what you have written, take 2 minutes, then we will discuss once again. So tell me, what are all the points we have focused on development of hindbrain? Your hindbrain is also called as Rhombencephalon, and it has 2 secondary cephalons, that is Metencephalon and Myelencephalon. For the development of hindbrain, we have to study about the Myelencephalon. If we take the cross section of Myelencephalon, similar to your spinal cord, we can see all the 3 cell layers, that is matrix cell layer, mantle cell layer and marginal cell layer, along with the neurocele part. Okay, and next Next, we'll concentrate on the dorsal and ventral part. The ventral part will grow rapidly compared to the dorsal part. As a result, only the ventral part will get expanded in size. So, your cavity of the ventral side will get compressed. Okay. So, next, what is going to happen? The roof plate present on the dorsal side, due to the Pontine flexure, it will get thin and it will also expand in both the direction. So, your alar lamina will be pushed towards the dorsolateral side. Okay. Next, once this changes takes place, the cells present in the alar lamina will start moving in both the direction, that is in your next layer, that is the marginal cell layer. Okay, whichever the cells or the group of cells that move towards the downward cells will going to give rise for the inferior olivary nucleus, and the group of cell that moves in the upward direction is going to give rise for the Pontine nucleus. And this expansion of cell from the Pontine region to your medulla oblongata region is called as check it once. Okay. Next, we have the formation of functional components. In the spinal cord, we have seen only about the general columns and it will give us 4 functional components. But in your hindbrain, that is in the part of medulla oblongata, totally we can see the 7 functional components. In the alar lamina part, we can see the 4 sets of columns, and in the basal lamina part, we can see the 3 sets of columns. I have written all the names, note it down. Any other doubts? So, in the next session, we have to study about the each and every column, like for which area of our body it is going to supply, or the nucleus and the nerves related to this part. Okay. So, I'll just tell the nerves name here. To your hindbrain functional components part, 5th, 8th, 9th, 10th, 11th and 12th cranial nerves are related. Okay. 5th, 8th, 9th, 10th, 11th and 12th cranial nerves are related in this area. We have to study about them, like individually we have to study. That we will discuss in the next session. So, which one is the 5th cranial nerve? Can you name them? Trigeminal. Okay. What about the 8th cranial nerve? Vestibular one, right? 9th one? Glossopharyngeal. 10th? Vagus. 11th? Spinal? Accessory. Okay. What about the 12th one? Hypoglossal, right? Okay. We'll study about that in the next session. I have added the notes, but only after completing the cranial nerves region, and also we have to study each and every component or the columns, which part of our body it is going to supply. Okay. After that, I'll discuss about these notes. Okay. We'll finish the session. Thank you, everyone.